And here we go.
Okay, awesome. I guess we can go ahead and get started. Hi, everyone, and welcome to Decoding Horse Health. It's a webinar series presented to you by EquiSeq. I'm Madison Sanders. I'm your moderator for today's discussion. And by way of introduction, I'm on the board of EquiSeq, and I'm also the CEO and founder of Elite Equine Marketplace, which is an interactive map-based hub that connects riders, trainers, and equine professionals with premium horses, quality equipment, boarding barns, services, niche services, and career opportunities. So goal is to make it easier than ever to buy, sell, and network with confidence. I'm also a lifelong hunter jumper rider myself. So I've seen firsthand the importance of supporting horse health at every level, from training to performance to nutrition and overall wellness. And today we're joined by two incredible experts. Paul Szauter, he's founder and CSO, Chief Scientific Officer at EquiSeq, and Kirsten Dimmler, who has just completed her PhD under the direction of Molly McHugh at the University of Minnesota. So we'll give them a chance to share their expertise, and then we've saved about 10 to 15 minutes at the end for audience Q&A. You all will be muted during the discussion, but please drop any questions that you might have into the chat as long as that chat is working, and we'll address as many as we can at the end, or we'll open it up and allow you guys to talk at the end if for some reason the Q&A is not working. But before we dive in, we want to be transparent about potential competing interests. As mentioned, I sit on the board for EquiSeq and I receive equity compensation for that. Paul Szauter, he's also a board member, of course, at EquiSeq, and he receives equity compensation for his work there. And Kirsten Dimmler is on the scientific advisory board. So she has received equity compensation for her work, leading to one of EquiSeq's patents, actually. And she's also impressively developed intellectual property for the University of Minnesota. But with that, let's give our guests a chance to introduce themselves. So, Paul, could you tell us a bit about your background and what got you started at EquiSeq?
Thank you, Maddie. So the first thing to say is I'm a recovering academic. I got my PhD in genetics in 1980, and I had a series of academic jobs as a Drosophila geneticist. And in 2000, I went through a career change and joined the Jackson Laboratory, which is a leading center in mouse genetics. And I worked there in genomics and bioinformatics. And in 2011, I moved to New Mexico for a position at the University of New Mexico, and I left UNM in 2014 and founded EquiSeq in 2015.
So, what made horse genetics an interesting opportunity for you?
Well, I've been fortunate enough to see a revolution in my field during the course of my career. The Human Genome Project dropped the cost of sequencing a human genome dramatically. So, if you had shown up at the completion of the Human Genome Project in 2002 and said, I'm very rich, I want my genome sequenced, it would have cost you $100 million. And in 2014, 12 years later, it was $1,000, and it's cheaper now. So, that's 5 orders of magnitude in 12 years. And that project produced an enormous volume of public data that's accessible to anyone, anywhere in the world, for free. And horse is an interesting opportunity because there's almost a total lack of federal funding for equine research. So it meant that a small company and a small group of people could make a real contribution to horse genetics.
Yeah, thank you, Paul. Kirsten, tell us something about yourself and your work over the past few years.
Yeah, definitely. So first, thanks for having me here today. I'm really excited to talk to everybody about RER. So my name is Kirsten Dimmler, and first and foremost, I've been a lifelong equine and animal enthusiast. I earned my bachelor's degree in biology from the University of New Mexico in 2018. And this is also where I met Paul and first got involved with EquiSeq. At the time, one of my own personal horses was affected by muscle disease, which led to me and Paul talking. And I learned that EquiSeq just happened to be based in my hometown. So it was kind of a serendipitous encounter. I ended up spending about 2 years working as a bioinformatician at EquiSeq. And around this time, I was also working as a veterinary assistant at a mixed animal clinic. So we did see horses there. In 2021, I began my PhD at the University of Minnesota under Dr. Molly McHugh, and my research there focused on furthering our understanding of the biology and genetics of recurrent exertional rhabdomyolysis. I worked in specifically standardbred and thoroughbred racehorses, and my work also included developing a genomic prediction model for RER, which I think we'll talk about a bit later.
Yeah, it's such an interesting path into the field, especially coming from both the clinical and genetic side, right? So I'm curious, how did working directly with horses and in a veterinary clinic shape the way that you approached your research?
Yeah, I think I've been really fortunate to see horse genetics from multiple perspectives. So, working with my own horses in a vet clinic and now as a researcher. And I think that combination has really shaped how I think about research. It's made me very focused on the practical side of things, such as how results will actually be used in real world settings. So I'm not just asking what's biologically interesting, but also will this be useful and how will it translate into practice?
Yeah, thanks, Kirsten. Let's turn to our subject for today, RER. So, what is RER from a basic perspective?
Yeah, so RER stands for Recurrent Exertional Rhabdomyolysis, which is a complex episodic neuromuscular disease primarily affecting racehorses, but it is found in other breeds. So we can kind of get an idea of what it is by looking at the name. So recurrent means that episodes of clinical signs are occurring repeatedly on a regular basis. Exertional means that episodes are triggered by exertion, for example, racing. Rhabdomyolysis refers to a rapid breakdown of muscle tissue, and this results in outward signs of stiffness, pain, sweating, and myoglobinuria, which is a coffee-colored urine that contains the protein myoglobin. So RER is a disease in which affected horses respond to exercise with episodes of muscle breakdown, even when the exercise is appropriate for the horse's current fitness level.
How common is that?
Different research groups have found varying prevalence, but we believe that about 5 to 10% of thoroughbreds and 10 to 25% of standardbreds show clinical signs. It's hard to be precise about the numbers because there's really no gold standard test to give a definitive diagnosis. Most of the clinical signs of RER are subjective, so things like muscle stiffness, pain, sweating, reluctance to move. So individual horse owners and also vets differ in their ability to identify the clinical signs. And also, individual horses differ in their presentation. So some horses are more stoic than others, and RER also varies a lot in how severe it is in different horses. So there are a lot of factors here that make it really complicated to diagnose.
Yeah, understandable. So, if there's no gold standard test, how is a horse diagnosed?
Generally, a vet will examine the overall clinical picture. So, does this horse show a pattern of episodes induced by exercise? What sort of diagnostic tests were performed? One thing that's commonly done is regular blood work to measure levels of creatine kinase, or CK, in the serum. CK is an enzyme that's found in muscle, and as the muscle tissue breaks down, this enzyme is released into the bloodstream. There's always a baseline level of CK in blood, even in healthy horses, because there's always some muscle breakdown in response to normal activity. But in a severe episode of RER, the level of CK can increase by nearly 100-fold. I should also note that elevated CK is not a definitive diagnostic measure. It's not uncommon for healthy horses to have elevated CK after exercise, and elevations in CK in RER-affected horses are sporadic. So a single serum sample can easily miss the elevation, or it can be lower than it would otherwise be.
Yeah. So, what does a vet do for a severe episode?
The most dangerous part of a severe episode of RER is the possibility of kidney damage. So during these severe episodes, myoglobin, which is a protein found in muscle cells, is released in high quantities into the bloodstream when the muscle cells burst. Myoglobin is a bulky molecule that can block blood flow to the kidneys and cause permanent damage. Additionally, certain electrolyte levels, like potassium, which is also found inside the cells, can become very high, which is dangerous to the heart. So to combat this, vets can administer large volumes of IV fluids to flush the system of these dangerous substances. These episodes are also extremely painful, so pain control is also a vital part of treatment.
Yeah, absolutely. Paul, have you ever seen a horse having a severe episode?
Unfortunately, yes. I attended an endurance event that was mostly Arabians, and there are spotters along the course that can pull a horse that's showing signs of lameness. And these are sent to a group of observers who evaluate the horse's movement and send the concerning cases to the vet trailer where we were stationed. So we saw the worst of the worst. And one horse was very stiff and in obvious pain, and the urine looked like coffee. CK levels are normally in the low hundreds, but in this horse, they were over 20,000, which is the upper limit that could be measured using the field instrument in the vet trailer. The vet spent 2 hours putting 30 liters of saline into the horse until the urine was clear, and the bill was $1,000.
Yeah, wow. Kirsten, do you have anything to add to that?
Yeah, so while severe episodes like Paul is describing do cause myoglobinuria and can be life-threatening, I think the most common presentation is really more subtle. A lot of affected horses never get myoglobinuria and instead just have those more subtle signs of muscle stiffness, pain, reluctance to move, and sweating that generally resolves without veterinary intervention. So RER exists on a spectrum of severity, both in terms of the severity of episodes themselves and the frequency of episodes.
Okay. So the horse Paul described was an Arabian, right? Does that mean RER is found in multiple other breeds, or more specifically, Arabians, thoroughbreds, standard breds, like we already discussed?
RER is absolutely found in other breeds, including the breeds you mentioned, but also in breeds you wouldn't expect, like ponies. Racehorses like thoroughbreds and standardbreds are just especially useful for studying RER because they're regularly subjected to intense exercise, both when they're training for racing and when they're racing. And the conditions they're kept in on the racetrack also can trigger RER signs. So it's very likely that many non-racehorse breeds, especially those that are closely related to thoroughbreds, have a genetic susceptibility to RER, but they're just never subjected to the conditions that trigger episodes.
Right. Yeah, that makes total sense. So how does RER differ from other forms of exercise intolerance?
Yeah, RER presents very differently from other forms of exercise intolerance. So first, RER is highly episodic. So when affected horses are not experiencing an episode, they're typically physically normal and show little to no signs of muscle pain. This is compared to other equine muscle diseases like PSSM and MFM, which tend to have clinical signs that fluctuate in severity, but are always present to some degree. MFM is also considered a progressive disease that worsens with age, whereas RER is not considered progressive and generally remains stable throughout the horse's life, depending on the management and the conditions that the horse is kept in. Another difference is that PSSM and MFM often involve muscle atrophy and weakness, while horses with RER usually don't show these signs. In fact, some studies have found that RER affected horses win races more frequently and earn more over their racing careers than unaffected horses. So this might help explain the relatively high prevalence of RER in racehorse populations. Lastly, RER is most likely not a primary muscle disease. Instead, its pathophysiology involves multiple biological systems in the body, including energy metabolism, calcium regulation, mitochondrial function, stress response, and many others.
Yeah. So you said earlier there's no gold standard for a diagnosis of RER. What about a muscle biopsy? Can that detect it?
Muscle biopsies are usually not recommended for diagnosing RER because the muscle tissue from affected horses usually only shows non-specific changes. The most common finding in muscle biopsies is an increased number of centrally located nuclei within muscle fibers, and this just indicates increased muscle regeneration. So likely we're seeing this because we know that in RER, the muscle is repeatedly breaking down and having to rebuild. This doesn't actually tell us what's causing that to happen. So this also supports our understanding that RER is caused by changes in many different systems rather than being considered a primary muscle disease.
Yeah, okay. So could it be inherited?
RER is definitely inherited. So it's considered a complex genetic disease, and that means that it's not inherited in a Mendelian manner where a single gene is responsible. Instead, susceptibility is determined by interactions between many different genetic mutations, as well as environmental factors like the diet. The genetic contribution to complex diseases is measured as heritability. And this represents the proportion of variation in disease risk that is attributable to genetics. So the heritability of RER is estimated at approximately 40% in thoroughbreds and standardbreds. And it's thought that about 600 to 700 different genetic mutations contribute to the overall risk. So in simplified terms, we could kind of say that RER is 40% genetic and 60% environmental, which we consider kind of a moderately genetic disease.
So let me step in here for a second and say a couple examples might help horse owners understand that idea. So albinism is a classic Mendelian trait. So mutations in a single gene are responsible for albinism, and the appearance of an albino is not affected by other genes or by the environment. And height in humans is a complex trait. So some single genes strongly influence height, but overall in the human population, the height of individuals of a similar age under similar conditions will show a bell-shaped or normal distribution. So height is determined by the interaction of hundreds of genes and the environment.
Yeah, going further with that height example, the heritability of height in humans is estimated around 80%. So again, this can mean that how tall a person is 80% due to genetics and 20% due to environment, like childhood nutrition. So we can compare this to RER, where the condition is 40% genetic and 60% due to the environment, like diet and exercise.
I want to interject again and say that as the environment changes, your estimate of the genetic contribution would change. So under conditions of perfect nutrition, much of the human variation in height would be attributed to genetics. And under conditions where nutrition is not optimal and there are other environmental factors that are severe, the environmental factor would seem to be larger than the genetic factor, right? So it also depends on the environment, how bad the environment is in which the individuals are being raised, right?
Yeah. So would you say, does that truly validate that the development of symptoms of RER is influenced by diet and exercise?
Yes. So diet and exercise are known environmental factors that influence RER. So many studies show that feeding a high-fat, low-carbohydrate diet is helpful to reduce the incidence of episodes. And on the exercise side, horses prone to RER benefit from a more prolonged cooldown period after being exercised and before being stalled.
Yeah. So are there any sort of supplements or drugs that can assist in that sort of treatment?
There is one drug. It's called dantrolene sodium. This is a ryanodine receptor antagonist, and it was originally developed to treat malignant hyperthermia. In RER, we use it as a prophylactic medication, which means that we give it before the horse exercises, and it's meant to prevent an episode from occurring. Once an episode is already underway, dantrolene doesn't really help. Dantrolene does have some side effects and is a fairly expensive drug, so usually it's only used for horses whose episodes can't be controlled with more conservative management, like changing the horse's diet or exercise routine.
Okay. Kirsten, you defended your dissertation in January this year, so very recently, and you've given us an overview of what is known about RER at this time. Can you summarize the work that you did over the past 4 years in a way that makes sense to horse people, say more in layman's terms?
Yeah, absolutely. I can give it my best shot. So there were two goals of my PhD research. The first goal was to create what's called a genomic prediction model for RER in racehorses. So you can think of a genomic prediction model similar to how weather is predicted in forecasts. So in weather forecasting, meteorologists use data like the air pressure, temperature, humidity, and other things to predict the likelihood of rain or clouds for the following days. This is similar to what I did in my research, where I used the genetic makeup of a cohort of racehorses as the data to predict the likelihood that they were affected by RER. So to do this, I started with a set of 32,000 genetic mutations that we believe are related to RER. And I think this probably seems like a huge number of mutations compared to how most horse owners typically think of genetic diseases. And this is because RER is very complex. We think that there are at least 600 to 700 mutations that are directly involved. But we knew that not all of these 32,000 mutations that we started with were probably important. So my task was to figure out which mutations were actually useful for predicting RER and to finalize that genomic prediction model. And hopefully that model will eventually become a commercial test for RER that could say, for example, that an individual horse has an 80% chance of developing RER in its lifetime just based off of its DNA. So during this work, I discovered, using my final model, that there were about 30 to 100 different mutations from that original 32,000 that were especially useful for predicting RER. And my final model predicted whether a horse had RER correctly 70% of the time in standard breds. This number might seem low, but it's actually about what we can expect from a model like this. Like we talked about before, RER is about 40% genetic. So my model used very few environmental factors, so we're pretty much relying completely on genetic information to predict RER. Since RER is only moderately genetic, there's no way to really get to 100% accuracy using only genetics. You'd have to include more environmental factors. Generally, for a moderately genetic disease, 70% accuracy is pretty good. And we do have plans to continue to refine this model in the future. And while we're eager to offer a test like this to horse owners, it's important that we're thorough, ensure that the model is clinically useful before we can make it available publicly. The second goal of my research was to better understand the biology of RER. So this means to get more insight into what's actually happening in the body that ultimately results in these episodes. To do this, I looked at the functions of genes that contain genetic mutations that were more common in horses affected by RER compared to healthy horses. And one of the most interesting things we found was that the excitability of the central nervous system may play a really major role in RER, which is not something that is heavily discussed in the literature. We found that several genes involved in calcium regulation within the nervous system were associated with RER. And this is important because muscle contraction is actually a really complex process that involves both the nervous system and the skeletal muscle system. Signals from the nervous system trigger the release of calcium, which then initiates muscle contraction in the muscle cells. So if cells in the nervous system are more easily excited, this could lower the threshold required to trigger muscle contraction, which might contribute to horses being more prone to episodes of rhabdomyolysis.
Yeah, so for riders or trainers who might not be familiar with the term rhabdomyolysis, is that what people commonly refer to as tying up?
In short, yes. So most racehorse trainers that I worked with refer to episodes of exertional rhabdomyolysis as tying up. But I think importantly, I've also found that in other fields of horse ownership, like Western sports or hunter-jumper, people also refer to the fluctuations of severity in other muscle diseases like PSSM or MFM as tying up. So these diseases can cause a similar outward presentation of signs, but they are biologically distinct.
So can I ask again, in human patients, where we know a lot more about human biology and genetics, what's the condition that most closely resembles RER? And what does that tell us?
Yeah, that's a really interesting question, Paul. So I would say that McArdle disease is probably the most similar condition in humans. McArdle disease is a Mendelian genetic disorder where patients lack an enzyme that's involved in generating energy from muscle cells. because the cells can't produce enough energy during exercise, patients experience muscle pain, exercise intolerance, and episodes of rhabdomyolysis. But I should also say there are a lot of differences between McArdle disease and RER. The most major being that McArdle disease is caused by a single mutation, while RER is caused by many. So I think it's really difficult to get a great comparison in humans because the muscular system of horses is actually really unique. Through thousands of years of selective breeding, horses have developed several genetic adaptations that affect how their muscles function overall. So without getting too far into the details, horse muscle cells are uniquely genetically primed to generate large amounts of energy, contract very strongly, and recover very quickly compared to other species. So these adaptations are what make horses such incredible athletes, but they may also predispose them to unique muscle disorders.
So it's interesting that you compare RER to McArdle's in humans. So I'm a little better acquainted with that than I used to be. So the McArdle disease, the affected enzyme encodes is a cytosolic enzyme that breaks down glycogen in muscle. And there's also a lysosomal enzyme in muscle that appears to be more important. And I'm up to date on that because we did a recent blog post on a drug for PSSM, and people want to know more about that, can look into it. But there's another human disease with exercise-induced rhabdomyolysis that I think could be compared to RER, and it's called CP2 deficiency. So that's a defect in carnitine palmitoyltransferase 2. That's an enzyme required for fatty acid oxidation, not carbohydrate metabolism. And so there's no evidence for a disorder of primary carnitine metabolism in horses. I think it's interesting the similarity of symptoms of both of these monogenic human diseases to RER is interesting. It tells us something about what's wrong. Both disease states affect energy production in muscle. McArdle disease affects glycogen catabolism, and CP2 deficiency affects fatty acid oxidation.
Yeah, that's a really good point. There's definitely evidence that both glycogen metabolism and fatty acid oxidation are major factors affecting RER. The difference is that those human diseases have a very clear single cause, where RER doesn't really seem to work that way. It's not one pathway going wrong, but more subtle changes across multiple systems, including majorly metabolic systems, like you mentioned, that add up to increase overall risk. I think it's also interesting that you brought up glycogen metabolism. I think we're so primed to think of glycogen with horses, mainly because of PSSM, but research into RER actually shows that fat metabolism may be more important for RER. It's possible that horses with RER are able to utilize the energy and fat at a much higher rate than healthy horses can, or even that RER horses are unable to access the energy and carbohydrates as well. So because of this, we actually believe that the high fat part of the diet is more important than the low carbohydrate part. So in short, it's less important to avoid carbohydrates in these horses and more important to add lots of healthy fats into the diet.
Yeah. So from your perspective, working directly with horses and horse owners, how is what you've learned or what we've learned about RER so far affected the way that racehorses are managed, for instance?
Yeah, when I was at the racetrack doing lots of sampling, I noticed that racehorse trainers tend to use a mix of anecdotal experience and scientific information to manage their horses. So most trainers I worked with were very aware of RER, and each sort of had their own way of dealing with it. I really commonly saw that horses were fed those high-fat, low-carbohydrate diets. That's one of the most well-supported management strategies in racehorses. Some trainers also avoided feeding alfalfa because of its calcium content and added electrolytes to the horse's water. And I think this likely comes from the understanding that calcium signaling plays a role in RER. But dietary studies haven't really found evidence that calcium intake itself affects RER clinical signs. Almost all of the trainers I worked with incorporated longer cooldown periods for susceptible horses. So after the horses were exercised in the morning, usually they're just hosed down and put back into their stalls. But when the trainers knew a horse was susceptible, they would walk the horse around for much longer. before hosing them and stalling them. I also noticed that a lot of trainers knew that their mares, particularly the mares with more nervous temperaments, seemed to be more prone to RER. So I remember one horse in particular. She was a thoroughbred mare that I met while I was sampling horses at the racetrack. I was about to go into her stall to sample her, but her trainer stopped me. He said that she had severe RER. He could barely control her tie-ups, but that she was also incredibly reactive and could be dangerous. So this would have been a really valuable horse to have in my data set because it's one of the clearest presentations of RER that I ever saw. But I wasn't actually able to ever sample her because she was just so reactive. You couldn't really predict what she was going to do next. But it was a really interesting case because it illustrated in real time something that I read about constantly during my PhD, which is that mares are much more likely to have RER, especially those that are nervous. So I think stress management is a much overlooked aspect of managing RER.
Yeah, talking about reducing stress, as a hunter jumper rider, I bring my horses to shows. So we haul them a long way sometimes and we get them settled into a new stall and new show ring, right? And I know that causes stress. So I can't even imagine the racehorse world. Any recommendations on reducing stress in these types of environments?
Yeah, so I think one way that horse owners can manage this stress is through training and enrichment. So on the training side, I think it's really important to do a lot of positive conditioning to the environments they're going to be in. So in particular, trailering is really important because trailers are especially difficult for horses that have RER, not only because they're stressful, but because the horse's movement is really constricted in a trailer and they're using their muscles in kind of unusual ways while they're trying to balance with the movement of the trailer. So this can put a lot of stress on them, and it really helps if they're really accustomed to the trailer and comfortable in it. So at least they're not stressed out while they're doing all of those muscle movements. So when they're actually at shows, or just in general, I think that one thing we can do is provide enrichment, which keeps the horse's mind active and keeps them happy and calm. So these can be foraging opportunities like hay bags in various locations or spreading the hay over a large area instead of just a pile. They also do make food puzzles that especially motivated horses might have fun with. Plenty of turnout is really important for RER horses and stress management for freedom of motion. Fitness level appropriate exercise on a regular basis. I mean, something that often gets noted with RER horses is that they can't skip a day of exercise. It throws them off completely. So regular exercise, but also like in a stall setting, there's jolly balls, mounted brushes they can rub up against, hanging toys and salt licks to keep that mind nice and calm.
So, Maddie, do you use any of those stress management techniques when you're trailing your horses?
Yeah, I do. I will say, not necessarily for hauling, but we do typically put salt licks or jolly balls in our horses' stalls when they get to the horse show. And one of my horses was recently on stall rest for an injury, and I hung a big stuffed animal in his stall. So some people have horses that need a buddy too, like a donkey or a mini as a companion. But yeah, before going into the show ring, we'll sometimes use Perfect Prep that has magnesium in it. It's show safe to try to quiet their minds. And that's kind of the magnitude of it, thinking about horse shows and rehab for my specific horses. I'm sure there's definitely other enrichment activities that I could consider that Kirsten just mentioned, though. I love that. But it does lead to another question of mine. And I just seen this done at a horse show. The horse is not quite right, and my trainer thought it could be a tie-up. So we took him down to the vet. We did some enzyme blood work to try to validate if he had a tie up during or, right after exercise within the hour. Is this something that can help determine if a horse has something like RER, at least early signs?
Sure. So doing blood work to measure serum CK levels can be really helpful as supporting evidence that a horse is prone to tying up, but it's not definitive. So in the best case, we would have a long history of blood work, both when the horse appears to be tying up and when it appears normal. So if we're seeing elevated CK after exercise and also when other clinical signs are present, this supports a diagnosis of RER, especially if the CK levels are really high. But like we talked about before, it can be easy to miss those elevations in any single sample. So that's why it's good to have a long history of repeated measures. And to complicate things further, other diseases like MFM or PSSM can cause similar clinical signs, like the muscle pain and stiffness. But they're also less likely to show CK elevations of RER. That said, it's not impossible for them to result in elevated CK. So sometimes PSSM or something like immune-mediated myositis, or IMM in particular, they can cause elevations of CK. They can even cause full-blown episodes of rhabdomyolysis, because rhabdomyolysis is really a general term. RER refers to repeated episodes of rhabdomyolysis, but there are lots of things that can cause rhabdo. So having just one episode of rhabdo doesn't really mean your horse has RER. So generally, we would need to see like a full clinical picture of each horse, including ruling out lots of other conditions to really feel confident diagnosing a horse with RER.
Yeah, so just a piece of the puzzle, it sounds like. Paul, anything to add on your end?
Yeah, I want to step back a bit and say this has been a great opportunity for me and Kirsten to share with our audience something about the scientific process. So I think a lot of people think that we go from knowing nothing to have a black and white answer that's absolutely true. And when Kirsten started her work, there was a basic description of the disease state and some ideas about management, and she's talking about 32,000 mutations, and she's got it down to, 100 or so in pathways that she understands are involved. Now, this isn't a black and white answer. There's not a commercial test yet, but it shows how an individual research project conducted over the course of 4 years can take a very intractable problem, genetic analysis of something that has only 40% heritability, and move it to the point where it's pointing toward a direction where it might be possible to develop a predictive commercial test. And that's passing the baton to the next set of researchers. And I think it's great for horse owners to see that there are some complex problems more closely related to many human diseases, and that everything that goes wrong with the horse is not the result of a mutation in a single gene.
Yeah, absolutely. Well, thank you, both Kirsten and Paul, for sharing such valuable insights today. I think there's been a lot of great information here, and I know many of our viewers will probably want to dive even deeper into this. So it looks like we're right at time. We've got about 20 minutes left, but I think we can open things up for audience Q&A. So it looks like the Q&A worked. Goodness, thank goodness. I see two questions right now. I can start off kind of just going down the line. So if the audience wants to add any further questions, now is your time to do so. I will go ahead and start off with the very first one by Kristen. She asks, my OTTB mare, off the track thoroughbred, has had multiple signs that she has RER. Yet she failed an initial test for RER at age 5, and she's tested negative in the EquiSeq panel for PSSM2 MIM muscle conditions. Yet she seems to do better following those suggested feeding and maintenance suggestions. What's the best way to manage the sudden explosivity that appears to be due to certain, I think she meant to say anxieties, anxieties?
Yeah, so I think this goes back to much of what we discussed. So this is a great example of a horse that may have RER and has a reactive temperament. So the best way to manage this is going to be the diet changes, so increased fat, lower carbohydrates, and like you mentioned, Maddie, there's the perfect prep that contains magnesium. So there is some evidence that magnesium can be used to calm down the nervous system. So that could be one approach that horse owners take to keep their horses calm. But I think I also really emphasize the holistic approach of managing stress and anxiety through the horse's environment. So I don't know what kind of conditions your horse is in, but if you can increase turnout and give more stimulation, more enrichment, I think that could be really helpful.
Yeah, looks like she added a follow-up on here too. What healthy fats do you recommend? My mare does not like flax. Trying chia now. My mare is incredibly sensitive to alfalfa and doing better now that I've removed that from her diet.
So I have to admit, I don't have a great recommendation on that. It's not really my area. I know people are commonly using flax or chia. I think corn oil is not really recommended because it's pro-inflammatory. But I used to use a feed by Triple Crown called Renew Gold, and I found that to be quite helpful. It's high fat, low carbohydrate. It worked really well for my thoroughbred, who was very old at the time, but he needed a lot of fat because he was a hard keeper, but he reacted very well to it. So I guess that would be my recommendation is Triple Crown Renew Gold.
Yeah, and I think equine nutritionists are really helpful to walk through that sort of process with you too. I use one at home in Virginia. She's been super, super helpful to me. So I can definitely reach out and put you in touch with her after this if you would like. But I do highly recommend for these sorts of questions. I have the same thing. I have some sensitive horses and I don't know these things. So I think finding a specialist in the equine nutrition space is recommended too.
Yeah, I think that's a really good recommendation because these diseases are so complex that often you need to be hitting it from multiple sides. And the professionals who are well trained in each of those different aspects of equine health are going to be the people you want to rely on.
Absolutely. Okay, next question is when do you view it as valid to get a muscle biopsy if RER is suspected by vets? EquiSeq showed no variants, but other disorders are not yet ruled out. This is another off the track thoroughbred mare. That's a common theme here. Very interesting.
While it's true that muscle biopsies are not particularly useful for RER specifically, RER is kind of a diagnosis of exclusion. So first, rule out other diseases, including MFM, PSSM2, PSSM1. I mean, not that thoroughbreds have that. But I mean, muscle biopsies are generally not much of an issue for most horses. They recover very well from them. I've seen it done several times. I've had a horse that had it. So, I mean, I would recommend just going ahead with it if your vet thinks it's a good idea. And that can help you rule out some other disorders and get you closer to, does this horse have RER or does it have a different muscle disorder?
Yeah, absolutely. Paul, anything to sort of
We
add
have a
to
couple
that?
more in comments. It turns out the way this works, you have to click something to expand them. But, I would say that when talking to horse owners and veterinarians about various aspects of horse disease, what I've been really impressed by is the complexity of the diagnostic tree that a veterinarian goes through. And I did hear a talk by an equine veterinarian where the first thing on the diagnostic tree is an evaluation of the horse owner. You know, are they of a nervous temperament? How good are they at observing the symptoms? Do they think that person is a good trainer and manager, right? And then the flip side of that is sometimes after a lot of diagnostic tests that cost a lot of money have come up with no answer, some veterinarians blame the horse owner. And I think that's one of the most tragic things that I've seen because horse owners, feel the pain that their horse has, and they want to know it's not their fault. And we're not very advanced in diagnosing horse diseases compared to human diseases. So if you can't get a convincing diagnosis and management plan for your horse, it doesn't mean you're a poor trainer. It means we don't know enough about equine science.
Yeah. This kind of leads into the next question too, is what can be done during a pre-purchase exam? I think this means tied to RER, right? If there's suspicion, it's, the suspicion that the horse has something like RER. This user mentions, I will be shopping soon. So, is there anything that can be done to detect before a purchase?
Yeah, so that's actually one of the really difficult things about RER is that it's difficult to diagnose. So I think the best thing you could get is as much information as possible from whoever you're buying the horse from. Try to get a good history because RER is mostly diagnosed based on the presence of clinical signs, so muscle stiffness, pain, reluctance to move, repeatedly over a large period of time. There's probably not anything you can do definitively. As we mentioned, like measuring CK one single time doesn't really provide a definitive yes or no. So this is one of the reasons that we're trying to develop this predictive model, because in the future, we'd like horse owners to be able to test any horse, even a brand new foal, and know the likelihood that it will develop RER in its lifetime. I mean, this is a big problem even on the racetrack, is trainers purchase horses at 2 years old and have no idea if they're going to have RER or not. And a lot of them do. And then they just have to manage it. So it's kind of something that in the future we hope to improve on. But right now, there's not going to be a definitive way to do a pre-purchase exam for RER.
Yeah. All right. I don't see any further questions here. So Paul and Kirsten, anything else you guys would like to add, or I'll leave it up to the audience for any last minute questions.
Well, I'd like to extend my congratulations to Kirsten once again for taking on a very tough problem and moving the ball forward quite a bit. And I think you can see, Maddie, from talking to both of us, that scientists are born optimists because if you weren't optimistic, you wouldn't believe that you could pry nature's secrets from her clutching hands. And I think you see an example here of the courage that it took to take on this problem. And I admire the work that Kirsten has done and the superior training that Dr. McHugh provided her at the University of Minnesota.
Yeah, thank
Yeah.
you. That means a lot to me. And it was a really interesting project. I really enjoyed my PhD and learned a lot.
Yeah, well, amazing. Well, thank you both so much. Thank you to the audience. We really appreciate your time here. I think this was super informative and valuable. So, that said, thank you all so much for joining Decoding Horse Health. And until next time.